All right. It's 3 minutes on top of the hour. I think we can start and hope that your colleagues will join along the line. So once again, I welcome you to MLB 203, Biochemistry II. Today, we will have some introduction to the course, and then perhaps we can take the very first topic in our plan. So right now, what you are seeing on your screen is the course outline. I think the semester is going to be for about 14 weeks or so. But a lot of times, I try as much as possible that the lectures that we have should be within a period of 12 weeks, taking into consideration the fact that sometimes anything can happen, some holidays, sometimes too. It's just that I feel you should finish in time, have about some 1 or 2 weeks before your examination starts so that you have enough time to prepare for examination. So I most of the times try as much as possible that my courses are within a period of 12 weeks. So let's go through the, well, somebody says you cannot hear, but in the beginning, I asked your colleagues. I think others can hear me. If you can't hear, you may have to check your settings, because at my end I can see that everything is clear. Okay, so the first thing we'll do is to have some introduction to the course, which is what we are doing now. And then we'll look at components of nucleic acids. What are nucleotides? What are nucleic acids? DNA, RNA. What are they made up of? Grace, Mackenzie, if you have anything, you can write in the chat so that I can attend to it. I can see your hand is up, but if you don't have anything to say, you can put your hand down. So the chat is active. If you have any concerns, you can write. You can write in the chat, and I will attend to it at the appropriate time. And at the end of the class, when I finish teaching, I will also open the floor. So if you have anything to say, you raise your hand. Then I will enable your microphone. You unmute yourself. You speak to the hearing of all of us, and then I can respond to whatever questions or concerns or comments that you have. Okay, let's continue. Then we continue to look at the structure of DNA, structure of RNA, look at some processes involved in the synthesis of proteins from DNA. So that is going to be DNA replication, DNA transcription, translation. Generally, these processes, particularly transcription and translation, are referred to as gene expression. That is the synthesis of proteins, which could become either some enzymes, some hormones, or some other, serve other functions in the body. Then, of course, if you are having processes of gene expression, then there should be a way of regulating these processes. So we'll look at what we refer to as gene regulation. We'll look at that in prokaryotes and also in eukaryotic cells, how these things, these processes of gene regulation occur. Then we move on to look at the structure of hemoglobin, which is responsible for the transport of oxygen from the lungs to the tissues and partly some of the carbon dioxide that is produced as a byproduct from cellular metabolism back to the lungs to be exhaled out of the body. Then we look at some abnormalities that can occur, what we refer to as hemoglobinopathies. Some abnormalities occur due to some abnormality in the structure of the prosthetic part of hemoglobin, which is the heme. And then what can happen if you have some abnormalities with the protein part, which is the globin. So we look at things like porphyrias. We also look at metabolism of hemoglobin. That is the synthesis of the heme, the prosthetic part. Once we talk about processes of transcription and translation, which is protein synthesis, then of course that takes care of the synthesis of the globin part, because that is how proteins are produced. So then you know how proteins of the hemoglobin are produced already, but then the synthesis of the heme, the prosthetic part, that non-protein part, we'll take a look at it. And then we will continue to look at some introduction to the endocrine system, looking at hormones, different types, the different kind, like different classes of hormones, starting from the hormones that are secreted from the brain, the hypothalamus, and then taking a look at hormones of the pituitary glands, the thyroid hormones. I mean, all the way going down to if you look at adrenal hormones, sex hormones, the different functions that they carry out and things like that. And lastly, we'll take a look at what we refer to as biotransformation. In other words, drug metabolism. Here we see it being depicted as a mechanism of detoxification, even though it's not all the time that biotransformation results into the detoxification of drugs. In fact, in some cases, biotransformation actually results in intoxication. We look at processes. So this is like when you eat food, the food goes through the digestive system to get digested, and then you have nutrients out of it. How about when you take in some chemical substances like maybe some medicines? Even if it was something which was toxic, you will be referring to it as a biotransformation of poison. So that will be in the field of toxicology. But in this case, well, we'll look at it in both ways, as if it's a medication or maybe it's a toxic as biotransformation. Where does that happen? Mostly in the liver. We'll take a look at the processes that are involved in such biotransformation. We may even look at it a bit as some biotransformation in the environment of substances as well. Now, you are already familiar with the assessment that your continuous assessment carries 40 marks. And then you have the exams carrying 60 marks. So continuous assessment will be, combination of all our activities over the semester, things like attendance, some assignments, tests, all of these things come together to give you your 40%. So that already you are familiar with. So this is the plan for the semester. I will share this plan with you after today's meeting. I will send it to the class rep, so the class rep will share it with you. And even after the lecture, the slides we'll be looking at as well, I will share it with the class, so you will have access to that. Over time, I will also look out for some book chapters and send to you. And what you can do is you use the slides as a guide to read some specific portions of the book chapters that I'll send you. Because, a book chapter can be about 30 pages, 40 pages, 50 pages. I mean, you don't have to go reading everything from the beginning to the end. You have to be like somebody mining for some specific information. And the guide to that is the slides. So you take the slide and see what we did in class, and then you just go specifically to the portions of the book chapters that I send you, because those ones are more elaborated, and then you can read on it and have a better understanding of what we discuss in class. Now, before we go on, I like to say that a lot of times I hear students complain about online lectures is difficult, is, so much different things that people say about it. But I think that it is actually an opportunity for you that each of you can sit behind a device, a screen, and can see clearly what we are talking about, compared to, say, having a large class and having a screen in front of the class that you have some people that are sitting behind and cannot see very well, and they cannot hear well what is being said. So I think it is rather an opportunity than, say, a disadvantage. No, it is rather an advantage to you, that if you actually pay attention to online engagements, you should do much better. Sometime in 2020, right after the COVID-19 pandemic, that was when ATU even hadn't gotten, they hadn't even gotten online teaching tools like Zoom and all of that. They hadn't gotten that. But at the time, there were still some form of restrictions to have, control over the number of students that will be on campus. So it was at that time that, so let's say that was even the onset of all of these online things, because some students will have to be on campus, some students will have to be outside of campus. And there were no online tools like Zoom that was procured by the university for lecturers to use. So lecturers will have to be innovative to see how best they can attend to the students at the time that they were not going to be on campus. And at that time, I remember I will record my lecture. That was more difficult because then I have to take some software. It was like talking to myself, record a lecture and send to the students. And there was a student in one of my classes that could not attend a lot of my lectures because she was sickle cell, and a lot of times she would be in crisis and be hospitalized, would be in pain. But she was one of the best students of that class. How? I mean, she came to me one day to say, thank you very much for your video lectures. And she showed me her notes. Said, look at my notes. If you look at it, you think she was in the class all the time and wrote everything, word to word. So it's about yourself, paying attention, sitting down, and doing your part. I refer to what I do. I mean, the work of a lecturer is to facilitate your learning, make it much easier for you. If you were just left for that, go and study biochemistry and come and write some exam, you wouldn't know what it is that you are actually going to pay attention to. So the lecturer is to just guide you. These are the things that you should learn. These are the things that you should pay attention to. This is how you should learn them. So I'm just to facilitate your learning, but in the end, you have to sit down and do your part. I will do my best to give you what you need, but you have to sit down and do much of the work. So it's just like we are on a journey, and maybe we plan that we want to travel to a place like Bolgatanga from Accra. Then I happen to take the lead, and you are behind. You haven't started from Accra yet. And I call you, and I'm just giving you some guide that when you are coming and you reach Konongo, pay attention to a certain curve there. When you get to this place, there is some pothole. Be careful. When you go to Techiman, do this. Yeah. I just give you such information, but in the end, you have to navigate your way yourself all the way till you get to the destination. So I'm just giving an analogy of what it is about a lecturer facilitating your learnings to give you a guide. But in the end, I will say, sit down. No complaints here and there. No, after all of this, the world is not going to treat you with some baby gloves outside there. So you have to toughen up and work, and no complaints here and there. Be open to new things. Don't just be stuck to some one thing which you have known and say you don't want to try any other thing. All right, so I hope that what I've said to you should encourage you to sit down and do the part of the work that you should do. All right, so if there's any question, if there's any comment, anything you would like to say and all of that, you can write in the chat, or jot anything, any concerns or any questions you have. You can jot them down as we continue, and then at the end you can ask, and I will attend to you. So I hope everything is clear from the beginning till now. If there is any best advice I give in my class, is that you should be patient with yourself. Perhaps it's not only just about biochemistry, but basically it may apply to every other aspect of your life. Be patient with yourself. Anytime that you try to do something for the first time, you are trying to study something for the first time and you are not understanding it, it can be frustrating. Sometimes perhaps even intimidating that can you do this at all? And the second time, you may be feeling maybe you are understanding, maybe not. But if you keep at it the third time, you'll be okay. By that time, you'll be having a better understanding of it. And biochemistry is such that you need to have that patience for yourself. If you understand one concept, it will carry you through because you will see it repeating over and over again. So I would say be patient with yourself and you'll be okay with biochemistry. All right, we can continue and go into the main business of the day. I said, if you look at a course outline, the first thing we have there is components of nucleic acids. So we can take that for today and then call it an evening till we meet the next time. Well, I think this is the second biochemistry you are doing in your life, because in the first year you did some, I think you call it biological chemistry. Yes, that's biochemistry. But what I notice in my experience is that even in the second year, a lot of you still have difficulty really, grasping what really it is that you are doing as far as biochemistry is concerned. You are still lost out there thinking what all this biochemistry is about. So what I intend to do is for us to have a very brief introduction to biochemistry, to have a very clear base for the whole course of biochemistry, so that at least I will hope that it will give you a clearer mind on what biochemistry is about. And then we move on to the main business. So let's see what biochemistry is about. Now, if you look at the word biochemistry, clearly it's coming from two words, which is biology and chemistry. In the secondary school, you did biology and you did chemistry, but you didn't have what we call biochemistry, which is a combination of the two. So you may be having some difficulty there because it will seem new to you. But actually what it is you are doing is, when you were doing biology, I mean, you knew biology to be the study of living things, simple. And you knew chemistry to be the study of chemical substances and then the interactions between these chemical substances. You studied these two things. And now you are having a combination of the two. So clearly, what really this means, biochemistry, only means that the interaction of chemical substances in living things. Because biology is the study of living things and the chemistry is the study of chemical substances and their interactions. So that here, if you have biochemistry, that is chemistry in living things. So chemistry in biological systems, as simple as that. But let's break it down stage by stage and have more detail from this. So I've broken these down into, say, 5 stages. So if biology is a study of living things, first thing you do at stage one biology is you do classification of the organism. So first of all, when you started to study science, even in primary school, you refer to living things and non-living things. That's how you grouped things: living things and non-living things. And the living things are what we call living organisms. So these are organisms. So if you take all the organisms, you start to study them, first thing you do is classification of these organisms. So is the organism a plant? Is it an animal? Is it fungi? So you did some classification into what you refer to as kingdoms, phyla, classes, order, family, genus, species. So you have this classification of all the living things, all the living organisms. At that stage for chemistry, what is chemistry dealing with? It's dealing with elements, because we're talking about chemical substances. And these chemical substances are elements. And the elements, there too, you did classification, what you refer to as the periodic table. So the classification you did there was to have these elements classified into things like, first of all, you have a classification on that periodic table into groups and periods. Then you start to look at what are alkali metals, transition metals, inert gases, halogens, actinides, lanthanides, etc. There was that classification of the elements at this stage. Then you move deeper down. In the case of biology, you now take a single organism, a single living thing. Remember, in the stage one, you were just classifying all the organisms, but now you take one particular organism and you want to look at its organization, how it is structured. And you look at the systems that make up the organism, what we are referring to as organ systems. So for instance, in human beings, you look at digestive system, you look at circulatory system, respiratory system, etc. What are these systems made up of? They are made up of organs. So you look at the organs, the heart, the stomach, the eyes, etc. All of these are organs. Then you look at what the organs are made up of. Say they are made up of tissues. What are these tissues made up of? They are made up of cells. And the cells, they are made up of organelles. So you look at the structure of the cell at that point, just to say that it's having maybe a cell membrane or a cell wall, and it's having a cytoplasm, which is having a fluid part, the cytosol, with some organelles in it. At that stage, that's the biology you did. How about chemistry? Now you take an element. What is the element made up of? Every element is made up of some atoms. So you start looking at the atomic structure. Say the atom is made up of a nucleus, and in the nucleus you have protons, which are positively charged, neutrons neutrally charged, or no charge, and around it you have some what you refer to as shells with electrons arranged around these nucleus. So you move further, stage 3, and at this stage for biology, you have gone advanced. So this is like a university biology, where you're now looking at structure and function. And particularly for you medical laboratory science students, what you refer to at this stage is anatomy and physiology, particularly when you're talking about the human body. And what is anatomy and physiology? Anatomy and physiology is the study of the structure and function of organs and tissues. So if you take the human body, for instance, if you take an organ like the heart, what's the structure of the heart? You start to look at the 4 chambers that the heart has, the ventricles, the kind of muscles that are in there. And then you say the function, how it contracts, how it relaxes, to transport blood across, the whole body in the circulatory system. You look at that. So anatomy is about structure and physiology is about function. Then in the case of chemistry at this stage, you start to look at the interaction between the elements. Or even before you start looking at that interaction between the elements, you start even looking at the interaction between the atoms of an element. So because you can have atoms of one particular element that are having some formation of bonds in them if a molecule of that element is diatomic, for instance. Say take maybe hydrogen, which is made up of 2 atoms. You talk about the bonds that are formed between these atoms. So you start talking about formation of bonds. Then, of course, you start talking about formation of molecules. Then, if you are having this formation of bonds between atoms of different elements, you start talking about formation of compounds. There can be some formation of complexes with bonds like some electrostatic interaction, hydrogen bonding, hydrophobic interactions. You start to look at all of these things for formation of complexes at this stage. Now, when you move down again to stage 4, biology and chemistry, now you have moved from the tissues to the cell, because now you're having an understanding that if the function of the tissue that you were talking about in stage 3 is actually coming from cells that make up these tissues, then of course the collective function of the cells will make up the function of a tissue. So you want to now see the structure and function of the cells. So you look at the cell, the structure, having a cell wall or cell membrane, the cytoplasm, the organelles in there. You start to study them, their functions, what they do, their structure. So if you take a mitochondrion, you start to see what's the structure of the mitochondrion, what's the function of it, what happens within a mitochondrion. So each and every organelle, you start to study their structure and their function. Golgi apparatus, endoplasmic reticulum, you start studying all of these things. And at this stage too, for chemistry, you start to look at interaction between the molecules. So you start to talk about chemical reactions. Here too, you do some classification. What type of chemical reactions do you have? Reaction types. You remember you did things like redox reactions. That is oxidation and reduction reactions. So you start to do such classifications, different types of reactions. You cause things like exothermic reactions, endothermic reactions. Then you also look at what are the reactants in a reaction? Which elements are reacting? And when they react, what do they produce? So what are the products? You also look at things like the energy profile of the reaction. So for instance, you study things like when reactants come together, they need what you refer to as activation energy for the reactants to move to a transition state. And then from the transition state, you now have a product. What are the conditions needed for the reaction to occur? Will the reaction occur in an acidic medium, or it will occur in a basic medium? So you look at biology and chemistry. At this point, when you move down, the next step, where now from the cell, you now move to the molecules, what you refer to as molecular biology. Structure and function of biomolecules. Because if you take the structure of the cell that we were looking at, that if you take let's say the cell membrane, for instance, you are now looking at what is the cell membrane made up of, and start looking at the molecules that are in the cell membrane. Okay, this is made up of some phospholipids with some proteins embedded in it, and perhaps some complexes of carbohydrates and proteins, glycoproteins, or glycolipids. That's carbohydrates and some lipids bonded together. Or you have some lipoproteins, proteins and lipids. So these are complexes between the molecules. So you realize that at this point of biology, you talk about molecules such as carbohydrates, proteins, lipids, nucleic acids. These are the biological molecules that you talk about. In all the biochemistry that you do, you realize that you've been talking about carbohydrates or proteins or lipids or nucleic acids, because these are the molecules that make up living organisms. Then, of course, once you are at this molecular level, you can now talk about the chemistry of these molecules. That is the interaction between these molecules. It means you start to talk about biochemical reactions. That is the reactions between these biomolecules. Together, all of these becomes what you refer to as metabolism. What's metabolism? Metabolism is a collection of reactions in living systems that is required for the sustenance of life of the living system. So you have here too, what are the reaction types that are inside the living organism? Obviously, there you have redox reactions, you have oxidation reduction reactions occurring. You have reactions occurring that are looking like acid-base reactions. So you have some acid-base interactions that can be occurring. You have reactants, what is going to react, and what is the product that will be formed. So that is chemistry you are looking at in the biological system. You take a look at the reaction energy profile as you do in chemistry. And this is where you start to talking about catalyzing the reaction. So reaction catalysis. You refer to that in biological system as biocatalysis. If you are running a physical reaction in the laboratory, you could apply some heat, give some energy to the reactants to increase the rate of the reaction in the laboratory. But that's not an option for biological systems, because you know the biological systems are made up of biomolecules. So things like proteins and nucleic acids, you know, when you increase the temperature, there will be what is referred to as denaturing of the proteins or the nucleic acids. You even have the breakdown of lipids. So you see there, you start to talk about enzymes, which are biological catalysts that will reduce the activation energy so that the reaction rate will be increased, because you need reactions in the biological system to occur at a rate such that the biological system will be sustained, because things have to happen in milliseconds. Because as I speak to you, I'm moving my hands, my eyes, I can hear even as I speak, my mouth, my jaw, you too. You are listening. Your eyes are moving. Your hands are moving. All of that. Everything that you are doing, it is being controlled by some reactions in your body. So chemical reactions are occurring in our body constantly. So I refer to biological systems as some complex chemical factory. So our bodies are complex chemical factories. So many chemical reactions occurring at the same time. In the biological system too, you look at what? Reaction conditions. So for instance, in your mouth, the salivary amylase that is there will be active at a certain pH. When it goes down to the stomach, it will become inactive because the pH in the stomach is different. A different enzyme will also be active there. So these are reaction conditions. So you can see clearly where biology and chemistry meet, and now you refer to it as biochemistry. So when you are doing biochemistry, you realize everything you are talking about is that you take a component, take a particular molecule. First, you want to look at the structure of the molecule. When you finish looking at the structure of the molecule, you now want to look at the function of the molecule. So throughout the biochemistry you'll be doing, you will see that you are always studying structure, function, structure, function. You take a biomolecule, what is it made up of? What's the structure? After you finish studying the structure, all the components in there, everything, then now, so what does this molecule do? You start looking at the kind of reactions that the molecule is involved in and all of that. So generally, this is what biochemistry is about. So I hope that now you have a very clear mind about biochemistry, so that you are still not in such a very vast vacuum of things and not really getting to grasp what really this biochemistry is about. So you pay attention and you see, you are always going to be dealing with things like carbohydrates, proteins, lipids, nucleic acids, every time. It is either you are studying structure or you are studying function. Right. Now, we can go in to the main business of the day, which is components of nucleic acids. So now you can see already we have taken one of the biomolecules, which is nucleic acids. And we are studying components. So that is what? Structure. When we finish studying structure, we move on and start looking at function. Now, nucleic acids are divided into two types. You have what you refer to as ribonucleic acid, which is RNA, and you have deoxyribonucleic acid, which is DNA. Perhaps even in first year, you might have touched on this. I think maybe even other courses, they have mentioned these things to you. I don't know if in the secondary school you did anything on DNA and RNA. And what are they? Generally, these are storage forms of genetic information in the cell. So you know that genetic information from parents to offsprings are transmitted by the nucleic acids. So this is what they are. Now, what are these nucleic acids made up of? The nucleic acids are made up of what we refer to as nucleotides. It's just like how you say that proteins are made up of amino acids. You also say carbohydrates are made up of monosaccharides. The same way, nucleic acids are made up of nucleotides. So what are nucleotides? We will now move and look at what nucleotides are. Now, a nucleotide is made up of three main components. A central 5-carbon sugar, which we refer to as a pentose. It's a 5-carbon sugar. And on the 5-carbon sugar, you have a nitrogenous base, what we refer to as a nitrogenous base. And we also have a phosphate group. So a nucleotide is made up of a pentose, 1, a nitrogenous base, 2, a phosphate group, 3. 3 components. From here, we will move to look at each of the components. But of course, a phosphate group, not really much to be looking at again, because a phosphate group is, you can see it clearly here already, it's an inorganic group that is coming from phosphoric acid. And just like all other acids, phosphoric acid group, this will give out hydrogen ions, and the phosphate group will be carrying negative charges. So nucleic acids in the aqueous environment in organisms carry negative charges, and the negative charges are carried specifically on the phosphate group, because it is the phosphate group that gives out hydrogen ions and then will be charged. So which means nucleic acids are charged. They are negatively charged in the body. Which is why when you go to the lab, practically, you go to the lab and you do things like electrophoresis. It is based on the knowledge that the nucleic acid is negatively charged. And so if you apply an electric field, then you can have the nucleic acids moving towards the anode. That is the positive terminal. And by that, you use that technology to separate DNA fragments based on their sizes. So really not much again to say about the phosphate group. So what to look at more in detail is the nitrogenous base and the sugar. So what we'll do is first we'll take the nitrogenous base and we'll look at it and say, what is that nitrogenous base? Then we'll take the sugar and say, what is this sugar? So let's go on. So the nitrogenous base. The nitrogenous bases are divided into 2 major groups. First group, purines. Second group, pyrimidines. Now, there are 2 purines and there are 3 pyrimidines. And what are the purines? The purines are adenine and guanine. And there's a note here that you find both adenine and guanine in RNA and DNA. Pyrimidines, you have cytosine, you have thymine, you have uracil. And there's a little note under the timing to say that you find timing in DNA only, and you find uracil in RNA only. Some of this information I think you already have, because I taught some students who were telling me they did something like this even in secondary school. Now, how do you distinguish between these things? Like, how do you actually study these things? Do you have to cramp these structures in your head and all of that? I think here, first thing, you want to distinguish between purines and pyrimidines. That's the first thing you should be able to do. Distinguish between purines and pyrimidines. Purines are made up of two rings. A six-sided ring that is fused with a five-sided ring. But pyrimidines are made up of only one ring, one six-sided ring. So quickly, when you see any question on nitrogenous bases and you see two rings, first thing that should come to you is this is a purine. When you see one ring, first thing that comes to you is this is a pyrimidine. Good. Now, if you distinguish that and you know that, okay, purine is made of two rings, so this is a purine. Now there are only two purines, adenine and guanine. How do you distinguish between adenine and guanine? And when you look at all these structures, there are some similarities. So for instance, if you take adenine and guanine, they look alike. But you see that on this carbon number 6 here, there is an amine group for adenine. You don't find that for guanine. Rather, you have a double bond oxygen on that same carbon number 6 for guanine. So that alone should make you distinguish between adenine and guanine. You see an amine group on carbon 6, that you have an adenine. Here you have a double bond oxygen, and then there is some amine group on carbon number 2. That is guanine having those two groups on it. So you can distinguish between adenine and guanine. Now, when you come to the pyrimidines, after you have distinguished it to know that it has only one ring, so it's a pyrimidine. Now, how do you distinguish between cytosine, thymine, and uracil? Here too, there are similarities. You see that all of them are having on carbon number 2 some double bond oxygen, double bond oxygen, double bond oxygen, all that the same. But you see that cytosine is the only one that on this carbon number 4 is having an amine group. The others, thymine, uracil, they don't have any amine group on them. So it means when you see an amine group, quickly, you know that that is cytosine. Now, when you take thymine and uracil, how do you distinguish between them? You see, both of them are having this double bond oxygen on carbon 4, double bond oxygen, they have the same. But you see that here on carbon number 5, you have a CH3, a methyl group for thymine. You don't find that in uracil. So that is the difference between thymine and uracil. You see a methyl group, quickly, that is thymine. And you see uracil is not having that methyl group. And then you'll be able to say this is uracil. So if you are having these structures in front of you and you are being asked to name them, this should be easy to do without mixing them. Okay, now I want you to pay attention to these hydrogens that are shaded blue. You see that in the purines on nitrogen numbered 9, you see there's a hydrogen shaded, adenine. For guanine also, a hydrogen shaded. You see hydrogen shaded on the nitrogen number 1 for all the pyrimidines. See those hydrogens that are shaded. Pay attention to those hydrogens because we'll come to them. You realize that these are the reactive parts of these nitrogenous bases. And so those are the parts that will be involved in bond formation. So keep that in mind. We'll come to it soon. Right. Now we've seen the purines and the pyrimidines as a nitrogenous basis. So let's move to the sugar, the pentose, that is found in nucleic acids. Here too, we have two types of the pentose in nucleic acids, two of them. We have what we refer to as ribose, and we have what we refer to as deoxyribose. So the ribose is what you find in RNA. So listen to the name. RNA is what? Ribonucleic acid. So that is coming from the sugar, the pentose which is present in RNA, ribose. So you have the name ribonucleic acid. In DNA, you have deoxyribose. Again, listen to the name. DNA is what? Deoxyribonucleic acid. So the name is coming from the sugar, deoxyribose. Now, what's the difference between ribose and deoxyribose? You see, when you take ribose, on carbon number 2, you see that there is a hydroxyl group shaded. In place of that hydroxyl group, for deoxyribose, it is only hydrogen. There is no hydroxyl group on carbon number 2. So think of it this way, deoxyribose, look, listen to the name, deoxy. So, just think of it as oxygen is deleted. Oxygen is removed without oxygen. So deoxyribose does not have a hydroxyl group. There is just a hydrogen. And ribose is having a hydroxyl group. So you have these two types of the 5-carbon sugar, the pentose, which is present in nucleic acid. Ribose is present in RNA, deoxyribose present in DNA. Right. Now, let's move a step further to now look at these components joining to give you the nucleotide. Now, remember I told you that you should pay attention to the hydrogens that were shaded in the nitrogenous bases. So have you seen? You have this hydrogen, which is shaded on nitrogen number 9. And this is adenine. And this is the ribose. You have this hydroxyl group that is interacting with that hydrogen. And you have a formation of water out from there. In the end, you have a bond which is formed between the nitrogenous base and the ribose. That bond we refer to as a glycosidic bond. That's a glycosidic bond. So you remember, in proteins, we say proteins are made up of amino acids. And what's the bond that is between the amino acids? Say peptide bonds. The same way, like say carbohydrates, they are made up of monosaccharides. And what's the bond that exists between the monosaccharides? They are glycosidic bonds. And this is a sugar. So usually sugar binding to another molecule, the bond which is formed between the sugar and another molecule is referred to also as a glycosidic bond. That's why this is being called a glycosidic bond. So the bond which is between the nitrogenous base and the sugar is a glycosidic bond. Now, pay attention to the fact that even though you are seeing that there is an interaction between a hydrogen on the nitrogenous base and a hydroxyl group on the sugar, do not confuse this with acid-base reaction. You may quickly think that, oh, it's a hydrogen and this is a hydroxyl group, as if it's an acid and a base that are reacting. This interaction is not an acid-base reaction. It is not. This is a condensation reaction. I mean, think of it like, the meteorologists will say condensation, referring to rain formation, water. So that's a condensation. Now, why is it not an acid-base reaction, even though you are seeing a hydrogen and a hydroxyl group? Remember, we said nitrogenous base. It's a base. So obviously, a base will not be giving out the hydrogen ions as acid will do. And the sugar is also not an alkaline. It's not a base. In fact, if you take any sugar, if you go to the lab, you can try it. Take any of these, even common sugar, for instance. Just pour it in water from some sugar solution and try to check the pH and see if it's going to give you a basic value. No, many of these sugars will have their solutions giving you slightly acidic to neutral. So you see some pH between maybe 4.55 up to about 7. So it's not a base. It's not a base. So this is a condensation reaction. Keep that in mind, that this is a condensation reaction. It is not an acid-base interaction in this case. All right. Then again, pay attention. This molecule that is formed over here is not yet a nucleotide. Remember, we said a nucleotide is made up of 3 components: a pentose, a nitrogenous base, and a phosphate group. There is no phosphate group here yet. You see, on carbon number 5, you see the hydroxyl group is there. No phosphate group is attached there. So this is not a nucleotide. It is a nucleoside. This is a nucleoside. So there's a difference between a nucleoside and a nucleotide. A nucleoside is just a sugar with a nitrogenous base. No phosphate group. Right. Same way as we just looked at with ribose, you can have a pyrimidine like uracil that will be bonding onto the pentose, also a glycosidic bond between that uracil and ribose to give you uridine. That's another nucleoside. Right. Now, when you have the phosphate group attached, you now have a nucleotide. Now here, the full structure which is being given you here is a common molecule you refer to, ATP, adenosine triphosphate, which is a form of cellular energy. It's a nucleotide, but it is not a nucleotide you find in nucleic acid. It has a different function as a form of energy in the body. Same way adenosine diphosphate. So you see, in nucleic acids, all the nucleotides are monophosphates. There's only one phosphate group. So that you have adenosine monophosphate, just this first phosphate group, which is attached onto the sugar, is what will give you that full structure of the adenosine monophosphate, which you'll find as a nucleotide in nucleic acids. But of course, here it's just being shown that you can have a nucleotide, which is a diphosphate, and a triphosphate. These are also nucleotides, but they have different functions. But all nucleotides that are in nucleic acids are monophosphates. Now, what's the bond that exists between the sugar and the phosphate group? It's being shown here as an ester bond, but specifically, this bond is a phosphoester bond. So what's an ester? So an ester will be having a carbon here in place of this phosphate group. In place of this phosphate atom, will be a carbon. So a carbon double bond, oxygen there, and then there will be an oxygen in between the two carbons. That will give you an ester group. Think you did something like this. Some of you, well, it depends on the school you went. Some schools have some very strong science base. You would have looked at a little bit of organic chemistry, and so you would have seen the formation of esters, particularly with glycerol and fatty acids. But I said specifically that bond is a phosphoester bond because in place of the carbon is the phosphorus atom. So it's a phosphoester. You come to understand when now we start to join the nucleotides, then we are referring to the type of bond which is formed between the nucleotides to give you a nucleic acid chain, say a fragment of a nucleic acid. What is the bond that exists there? So if you keep this one in mind as a phosphoester bond, when we mention the name of that bond, then you understand that yes, this is a phosphoester bond. Now, you see, we said a nucleoside is made up of just the sugar with the nitrogenous base bonded on it, no phosphate group yet, which means that we can say that a nucleotide is a phosphorylated nucleoside. So a nucleotide is a nucleoside that has been phosphorylated, on which you have phosphate group. That's what it means. You can say that. All right. Now, if we, summarize all of these, it means that if you take DNA, you have 4 types of nucleotides that will be present in DNA. What are the bases that are present in DNA? Adenine, guanine, thymine, cytosine. Now, when these bases bind to deoxyribose, they form their corresponding nucleosides. So you have the nucleosides being deoxyadenosine for adenine, deoxyguanosine for guanine, deoxythymidine for thymine, deoxycytidine for cytosine. Now, if these nucleosides, when you have these nucleosides phosphorylated to form the nucleotides, you will now have for adenine deoxyadenosine monophosphate. Yes, there's some 5' there. That is to tell you that the phosphate group is on carbon number 5 of the pentose. And you see the abbreviations to distinguish these nucleotides from the nucleotides that are present in RNA. You see there's a small letter D in front, which is to say it is deoxy. The ribose there is deoxy. So deoxy adenosine monophosphate. You see deoxyguanosine monophosphate, deoxythymidine monophosphate, and deoxycytidine monophosphate. These are the nucleotides that you find in DNA. When you come to RNA, remember in place of thymine, you have uracil. So you have adenine, guanine, uracil, and cytosine. The nucleosides there, so this time around, it's not deoxy. It is a ribose. The pentose there is ribose. So the nucleoside you have is adenosine for adenine. You have guanosine, you have uridine, and you have cytidine. So that's the difference between the nucleosides. And when you move on to those nucleosides being phosphorylated, you now will have adenosine monophosphate, not deoxy. And you also have guanosine monophosphate, uridine monophosphate, and cytidine monophosphate. So Vincent Akoli is saying that my line is not stable. I don't think it is my line because I can see my signal is very strong. Perhaps it is yours, not mine. In any case, after the lecture, I will give you the link to the video so you can watch it again as many times as you can, so you don't lose anything out of it. Okay. Now, apart from these nucleotides that we have looked at in nucleic acids, when you read biochemistry, you may come across some other kinds of nucleotides, which are not in nucleic acids per se, but it's okay to take a glance at them and know that they are also there. So we're looking at some, just like we saw that you can have adenosine triphosphate, adenosine diphosphate as some energy forms, cellular energy forms, but they are not in nucleic acids. The same way you have some cyclic nucleotides. So for instance, you often see something like a cyclic adenosine monophosphate. So you see the phosphate group, which is on carbon number 5, is forming an intramolecular a cyclic structure with that carbon number 3. So you see that the name there says 3', 5' cyclic nucleotides, because the cyclic structure is formed between carbon number 5 and carbon number 3 of the pentose. The base there is adenine. That's why it's a cyclic adenosine monophosphate. You see this structure as some second messenger when we move on to look at hormonal function. We'll come to see that some hormones require cyclic adenosine monophosphate as second messengers for signal transduction or signal transmission. You'll see that. Similarly, you see something like a cyclic guanosine monophosphate. It's also a 3', 5' cyclic nucleotide. These two activate some enzymes, some protein kinases, so which will be responsible for the transfer of phosphate group from one molecule to the other. I mean, generally, these are for the activation of molecules for the initiation of some biochemical pathways. You will come to see some of these things as you go ahead in biochemistry. You can have something like a 2', 3' cyclic nucleotides. Here too, you see the cyclic structure is formed between the carbon number 2 and carbon number 3 of the pentose, the sugar. And these, generally, they are some intermediates in the process of metabolism of nucleotides. So, nucleotides, just like all other biological molecules, undergo some recycling in the body. The biological system is not a static system, it's dynamic. And so you have some recycling, you have some cyclic processes that occur. And so you have some of these intermediates of nucleotides that form in the process of metabolism of nucleotides themselves. And these, upon these, you have some nucleases, some ribonucleases acting on them, breaking them down into their various components. And they can come back together again to form some new nucleotides again. So these ones too are there. You may see them along the line. Then you can also see cyclic dinucleotides. So this time around, you are seeing 2 nucleotides that are forming a cyclic structure. So, I mean, just take your time. Don't be intimidated by the structure. You first just look at, you take one sugar, and this sugar is having a base on it. And that base is what? That is guanine. And it's having a phosphate group. So that is one nucleotide. You take the second sugar, that sugar facing downwards is also having a nitrogenous base on it. And that nitrogenous base is what? That is adenine. And it's also having a phosphate group on it. And you see that these 2 nucleotides are bonded on the carbon number 3 for one of the sugars with that phosphate group, which is on carbon number 5 from the other. And here too, you have that phosphate group on carbon number 5 for that nucleotide bonding with carbon number 2 of the other nucleotide. So it's forming that kind of cyclic structure. Now, these dinucleotides, you will find them in some bacteria carrying out several functions responsible for the movement of the bacteria or the formation of some protective matrix for bacteria, which you refer to as biofilms, or even responsible for the ability of the bacteria to cause harm to its host, what you refer to as virulence of the bacteria. You may see some of these things as you go further in biochemistry. But these are not nucleotides that you find in nucleic acids. So you keep in mind that the nucleic acids that you find, the nucleotides that you find in nucleic acids are monophosphates. All right, so that will bring us to the end of this introductory lecture to our course. Now the floor is open. If you have any questions, any concerns, any comments, any contributions, you can raise your hand and I will enable your mic. Then you will unmute yourself and you speak. Alternatively, you can also write in the chat and I'll respond to it. So the floor is open now. I've been talking for over 1 hour. You can see it's 1 hour, 1 hour, 30 minutes plus. So I'll give the rest of the 30 minutes to you. So the floor is open. All right, so I see one hand is up. Let me see, let me